couple more organelles, and then we'll move on to some different topics. There are what are called energy generating organelles, and there are two specific types here. One I mentioned earlier is the mitochondria, the powerhouse of the cell. This organelle, shown over here, this is a real microscope image with the orange coloring, and this obviously here is an illustration. But the mitochondria, fairly complex little thing. I don't want to get into too much detail. It's another can of worms you open up if you try and talk about it too much. So really keep it very simple, and that is in the sense that the mitochondria is an energy generating organelle, and then what I want you to know is it generates ATP from glucose. So glucose is a sugar. Your body converts, obviously sugar can be used, I should say sugar is used directly to make ATP, but your body can even break things down like fats and proteins into sugar, which can then be used by mitochondria. So there's a lot of ways to create glucose in your body through a variety of food sources. And your body does that and then ultimately feeds the mitochondria with that glucose to make ATP. It can use other things besides glucose, but that's kind of its main energy source. So that's the mitochondria. If you want to know more, there's an interesting connection in evolution called the endosymbiotic theory. And there's just not enough time to talk about all this, but for anybody who's interested in evolution of eukaryotes, the endosymbiotic theory is a proposed mechanism by which eukaryotic cells evolved from prokaryotic cells. And interestingly, what it suggests, with a really high degree of certainty and lots of evidence to support this, what it basically suggests is that mitochondria are actually bacteria that emerged, or co-evolved rather, with the eukaryotic cell to create a new source of energy for the cell. So it's kind of an example of co-evolution between two organisms that went on to become one organism. And we actually see that the mitochondria looks and has all the features of a bacteria. So the theory is pretty sound in that this thing has all the features of a bacteria. It's just that it's no longer a separate functioning organism. It's now part of a eukaryotic cell, almost like a slave to the eukaryotic cell. And its main purpose is to generate extra energy. The same is actually true of the chloroplasts. These are both thought to be bacteria-like organisms that were kind of trapped within eukaryotic cells with the purpose of generating energy. The chloroplast is a different type of organelle in that it generates or rather it conducts photosynthesis. So it's a different type of energy generating process. So photosynthesis is unique to plants and algae. There's also bacteria that do this, but from a eukaryotic standpoint, these are both plants and algae. Remember, I compared those two together back in chapter 1. Don't really want to say too much more, but just recognize that these are the two energy generating organelles. Mitochondria makes ATP from glucose. Chloroplast generates energy through photosynthesis, which, if you're not familiar with that, which is where light, water, and CO2 generate glucose. And that's actually how glucose is formed on Earth. So, okay, moving on here. Ribosomes we mentioned in chapter 4. Talk about those again here. Remember, those are similar features in both types of cells. Ribosomes, protein synthesizers, we've already talked about that. This shows you a better structure, a better figure of the structure. Ribosomes are actually made up of two specific units. One's called a large subunit, the other's called a small. And they actually function when the two come together and almost sandwich together, kind of like hamburger buns. I always think of the ribosome as kind of like a hamburger bun. The only thing I really want to point out here is that while both cell types have ribosomes, there is a difference between the exact types that they are. So eukaryotic ribosomes are said to be what are called ADS ribosomes, kind of an arbitrary name that they just kind of made up based on some of the properties that they observed in the lab. So I don't want to get into why they call it that, but just recognize it's called the ADS ribosome. And prokaryotic ribosomes are called the 70S ribosome. So while they both have ribosomes, they are two distinctly different types. I like to compare them to two different types of trucks, like a Chevy versus a Ford. Chevys and Fords are both trucks. They both have four wheels. They both have a truck bed. They both have an engine. They both do the same thing, but structurally they have different components, and if you take them apart and tinker with the internal gears, they're just a little bit different. And that's kind of how ribosomes are. Do the same thing, largely look the same, but there are different structural components to them. this is only significant because one of the things we'll talk about later in the semester is how these types of subtle differences can lead to significant ways of targeting one type of organism or another. For example, we can use antibiotics that actually target the 70S ribosome and not the 80S ribosome. So drugs like tetracycline are actually specific to only targeting the 70S. It would almost be like having a virus that targeted the computer board of a Chevy truck and not the computer board of a Ford truck. You know, all trucks and cars have these computer chips built within them, right? So imagine a computer virus that you could insert into the motherboard of a Chevy, but not that of a Ford. It's kind of like how antibiotics work. It's a drug that only targets one part of the 70S in this example and doesn't affect the 80S whatsoever. So when you take a drug like tetracycline, you have this ability to target only the 70S and not the 80S. Okay, the next thing I want to talk about is what's called the eukaryotic cell cytoskeleton. The cytoskeleton is essentially a group of proteins that help kind of make up some of what I like to think of like the scaffolding and like the structural beams almost of the cell. So you can almost think about like a large building, and you have to have support beams, and you have to have reinforcement around the outside of the building. You have to have kind of this architectural design that allows the building to be supported on a large scale. So just to give you a few visuals, think about it like this. Here's some pictures of a building. You've got some of these cross beams, and you've got these designs that are meant to kind of help reinforce the shape of the building. Ideally, it helps it not collapse from within. You've got these key designs in beams and angles to kind of help redistribute the weight, kind of help make up some of the ability of these buildings to hold extra weight. Also, what allows the building to get larger. You know, you can't make a really, really large, complex building without a key set of designs that kind of allow that building to hold extra weight and support itself. So in a eukaryotic cell, you can think of it kind of the same way. Remember, eukaryotic cells are much bigger than a prokaryotic cell. And one of the things that happens as you get bigger is you have to get more structurally reinforced. So we don't really see this in the prokaryotes, and that has partly to do with why they're small and also how they can get away with being small. So if you make a really tiny little, almost like living in a tent, for example, or like a really small little shack, you don't necessarily have to spend as much time on the key structural reinforcement of that building. If you want to make a really, really large house or really big complex design in reference to a building or architecture, again, you have to spend more time setting up the support beams and kind of how all the weight is redistributed and how many people you can hold and that sort of thing. So that's kind of like what we're looking at here with the eukaryotic cell cytoskeleton. It's really kind of like an architectural design of the cell that allows it to be supported. It also goes on to do other things such as help with movement and transportation within the cell. So it goes a little bit beyond that. So it helps to anchor organelles. It helps to transport cellular vesicles. It helps to permit shape changes and movement. And that all is kind of part of within this idea of kind of creating the scaffolding and the structural support of the cell. Here you're looking at a figure out of the text. This is showing that figure we showed in the very beginning, the overview figure. And you're seeing here these little hair-like structures. These are all part of the cytoskeleton. This here and down here, these are fluorescent microscope images of cells in which they've highlighted just the cytoskeleton. So you don't see the nucleus, or you don't see rather the cell membrane, you don't see the other organelles, you see just the cell cytoskeleton, plus in this case you actually see the nucleus. So this blue is the nucleus, you kind of see where the center of the cell is, and then all around it here you see just the cell cytoskeleton. So you can see how this kind of permeates inside the cell and generates this kind of scaffolding that the cell can then kind of be supported around. Now there are three types. So just real quick, you want to know what are the three functions. You also want to know what are the three types. And each one is a little different. So you also want to know just a basic idea of what each one does. I'm not going to ask you real complicated, in-depth questions on this. I just want you to have a basic understanding of the three functions, the three types, and a basic idea of what each type does. So one type is called a microfilament, another type is called a microtubule, another type is called an intermediate filament. Each one is a little different. Microfilaments are these long kind of thread-like proteins, and these are found in the highest concentration around the cell membrane. So going back here, you can see the microfilaments. By the way, this is where biology gets frustrating, I know. They also refer to these as actin filaments. So actin filaments and microfilaments are the same thing. I apologize, I didn't have that stated here. But what they do is make up a lot of the scaffolding around the outside of the cell. Let's see here. You can see the microfilaments, called microfilaments here, and you can see here's the membrane in blue, and the microfilament is attached directly to that, meaning it makes up just around the outside of that cell. One of the big things that they do is they help the cell in reference to movement and contraction. Okay, real quick, I found this a part of a video I want to show you that talks about how microfilaments can actually be responsible for cell movement and contraction. I have to mute the sound because it's absolutely horrible quality and a very poor explanation. But let me just kind of walk you through just a real quick video on mute to show you kind of what's happening here. So here's a cell. This could be a eukaryotic cell of almost any kind. What we're going to see here is how the actin filaments, and they're called actin filaments in this video, again, same thing as microfilaments. We're going to show how they work. So what's going to happen here is that the microfilaments, the actin filaments, are going to push the membrane. So this is the membrane around the outside of a cell. We're going to push the membrane in one direction, and what that's actually going to do is actually move the cell in that direction. So it's going to kind of push it in one direction by the actin filament extension, and then we're going to see the back half of the cell kind of contract, and what we end up seeing is what's called cell crawling, in which the cell is pushed in one direction and then kind of contracted, ultimately kind of gliding it in one direction. So here you're seeing a part of the cell that's going to be involved in movement, and we're going to see here how the actin starts to actually concentrate. I'm not showing here, here's the actin, here's the filaments that make up around the outside of the cell. What we're going to see is that they're actually going to start to grow and extend and actually physically push the membrane with them. So here it's showing there's the actin growing, and what that actually does is it physically moves that membrane forward. So you're actually seeing movement take place in real time through the extension of the actin filaments or the microfilaments. So as they push forward, that actually causes the cell to move. So here's another view of that from the side. So this is like a single-celled amoeba type organism. And right here in red, this is where those actin filaments are extending, and it's going to push the organism in this direction. So it talks about how it anchors here. That's a little bit extra information. But here it shows that extending outwards. That's through the microfilaments or the actin filaments being pushed out. That physically moves the organism forward just a little bit. And then what ends up happening is some of the other actin filaments here inside are actually going to contract themselves and actually bend forward. So you kind of see an extension followed by a contraction on the back half. And this is done very, very quickly. This is obviously shown a very slow moving time here. So here's another part of that contraction. So this is all taking place in real slow time here, but organisms do this very rapidly, and they extend out one part of the cell, and then they contract the back half of that cell. Real quick here, this is when I pull up. You can see this in real time here. This is a real organism. So you can see how this would be extending forward and then contracting on the back half here. Hard to see all that. Life is really complicated, and this is taking place really fast. But that's actually what's happening on the microscopic level. You're actually seeing, this is kind of from the same source here, you're actually seeing those extensions followed by contractions that are taking place in real time. So that's actually one way that certain cells can go about moving, by using part of the cell's cytoskeleton. That's a pretty amazing thing that they do, which is to extend and contract, and that actually leads to cellular movement, which is usually referred to as cell crawling. Not all cells do that, but for a lot of cells that move, that's one way that they can go about doing that. Now, another key part of these cytoskeletal features is called the microtubules. These are long, hollow protein tubes, and those are shown here. Let's find just a slightly better figure of those here. Here we go. Usually show, here's a better one. Here we go. There's kind of a microtubule. You can see it has a hollow structure. Here's a cross-sectional view made up of a couple different proteins called tubulins. So these literally create almost like a PVC pipe type structure, which is hollow on the inside, and that then allows it to serve its function, which a lot of times is part of the overall shape, but it helps to transport substances and allows for movement of things in and out of the cell. It also is a part of the flagella and the cilia in eukaryotic cells that we're going to talk about next. So it actually helps to create a new structural complexity of the flagella that helps it move back and forth. So we'll see that here in a second. This is a really cool video, totally animated and a little bit dramatized, but just to give you an idea of how microtubules might actually be responsible for movement, and also actually how vesicles move throughout the cell. I want to show you this video. So this is a microtubule, and this is a cellular vesicle, one that we talked about earlier. So earlier I just mentioned how these just float across the cell, kind of like I showed here. So here's the vesicle, and we said they just kind of float from one side of the cell to the other. The truth is, they don't necessarily do this so magically. They're actually being drug along microtubules. So we just won't always show that detail because it's so complicated. You can't necessarily just put every piece of detail in every drawing and every figure. So to give a little more detail here and give you a better idea how that works, you actually have a microtubule that would extend throughout the cell, and here's the vesicle, and the way it moves is by these little proteins called kinesins, and what the proteins actually do is move along the microtubule. And they literally do so by activating these two little leg-like structures that physically carry that vesicle from one side of the cell to the other. This is real, by the way. This is not fiction. This is a real thing. This is actually what's called a motor protein. And the motor protein is being activated by energy, and it literally drags that vesicle from one side of the cell to the other. Very short little video there, but that's actually how things typically move about the cell. So when we talk about how it's responsible for cellular movement, really what it does is it acts almost like a little road by which vesicles can travel along with the assistance of other little proteins, in that case a motor protein. Life is really complex, and so that's just a kind of a little more detailed glimpse into how all that actually kind of works. The last type is called an intermediate filament. These are often found throughout the cytoplasm and often help reinforce some of the organelles and things like that. So mitochondria, for example, here are shown. This is a mitochondria, and you can see some of these intermediate filaments here kind of helping to make up the internal scaffolding and kind of help reinforce where these organelles go. Also might help kind of reshape some of the organelles throughout the cell. Okay. Next thing, we want to talk a little bit about the motility appendages for eukaryotes in terms of their extensions from the cell. In chapter 4, we talked about the flagella and the fimbriae. We also talked about, I misspoke, just the flagella for motility. Bacteria have only flagella for a motility feature. In eukaryotes, we see a very similar structure. Actually, it's very different structurally, but similar functional, I should say. It's actually called the same thing. So we have a tail-like structure here that is very similar to that of a prokaryote in function. So there's a couple different types here. Here's a tail. Here's a series of tails, all flagella. So it's similar in function. Similarities in that it's used for movement, extends out, and it moves in response to stimulation. We didn't really talk about that in chapter 4, but just to clarify, the tail is moved based on some kind of stimulation. They don't do so randomly. They wait till they have either a positive or a favorable stimulation, and then they either move towards it or away from it. So all that is similar. The key differences here is that it's significantly more complex. The eukaryotic flagella is said to have what's called a 9+2 arrangement, which gives it a more dynamic range of motion and makes it act like a true tail. We talked about the prokaryotic flagella, and I mentioned that it's very, very simple. It really kind of only moves in two directions. It either can rotate clockwise or counterclockwise, and it really only has a couple basic lines of movement. It can either kind of move straight, or it can kind of move randomly and reorientate. So it's very, very simple in terms of its actual ability to direct a cell with a certain level of precision. Bacteria kind of move clunky and random, and it takes them a long time to get anywhere. The eukaryotic flagella is much more complex. So this is a side view of the flagella extending out from the membrane. When you look at it inside, it's actually got these structures here, which are the microtubules, shown in a series, which is what we refer to as the 9+2. So here it's another figure showing the microtubules, what's called central and double microtubules. What you essentially have are 9 pairs of microtubules around the outside, or rather around the periphery of the flagella. That's where the term 9 comes in. And then you have 2 microtubules inside here paired together, and that's where the 2 comes in. So when we say it has a 9+2 arrangement, that is in reference to this organization here with 9 and then 2 in the middle. Again, very random name. They could have called it anything, but that's what they decided to call it. Now, the way this works, these microtubules will actually kind of glide past each other and allow the tail to move in a variety of directions. Okay, real quick, let's take a look at a real quick animation here on mute. This is going to show kind of how the flagella begins, how it extends, and then kind of how it moves up and down. So here we're looking at the beginning of the flagella formation. These are structures called centrioles. They're going to divide, and they're ultimately going to move towards the membrane. Now, these have multiple functions. Centrioles are used in cell division also, but here they serve towards the production of the flagella. So they anchor into the membrane, which now they're called a basal body, and they literally kind of extend outward. And so this becomes the beginning of a flagella, also cilia, which we'll talk about next. So here you're looking at the 9+2 arrangement. Here's the 9, here's the 2 in the middle. Now what you have here, these little white structures are called motor proteins. And what can happen is the motor proteins can actually slide down past each other on one side or the other. So at first they're nice and parallel, but what can happen is on one side of the flagella, the motor proteins slide down. What that does is it causes tension, which makes the flagella move like this. You're basically pulling this side like that, okay? Or you can pull the other side like that. And you can do this in any direction. So this is where you can get really all 3D dynamic motion. So when one side pulls, the flagella bends in that direction or the other direction or forward or backward. And that's how the flagella or the cilia are going to move. So here you can see that. Got it on mute. And it's going to start moving here in a second. Here we go. So here you can see as it starts to bend and move. So in this case, the flagella are bending down and then they're bending up, and that's all based on the motor proteins that are sliding past each other. And yes, you can come up with all sorts of jokes right now, and I'm sure that's really funny, but that's the idea. So you can get movement based on the sliding of these proteins back and forth, and that can happen in any direction, which gives it a very, very dynamic, three-dimensional sense of rotation. That may not make perfect sense, but hopefully it makes a little bit of sense. So really all I want you to know is the 9+2 arrangement and kind of how that gives us a more complex variety of motion. Now, we're not going to go through all of the different arrangements here like we did with the prokaryotes, because there's so many of them that it would take us a much longer amount of time. So there's a huge amount of variation as to how these flagella are arranged, and so we're just going to skip past that. Now, there's another similar structure called a cilia, which is a smaller version of a flagella and typically more numerous. So just go back here real quick. Here's a flagella, kind of a traditional single tail. Here are cilia. They're the same 9+2 arrangement, but they're just shorter and more numerous and stick out all around the outside of the cell typically, although I'll talk about some exceptions to that. So I like to think of flagella as more like one major, or maybe a couple major motors that is on the back of a boat. Think of like a flagella like this. Think of this as like a little boat in the water. Think of this as like one big motor on the back, and you kind of have one big mechanism of propulsion. Cilia, I like to think of like a Viking ship with lots of little oars on both sides. And here's a visual for you there. So kind of like how you have to have lots of little tiny oars all moving simultaneously. That's kind of how the cilia work. They all kind of beat in unison and kind of propel the organism forward, kind of like that. Whereas the flagella, again, is more like one big propulsion device at the back. Now, so cilia are used for movement. They're also used in some organisms for filter feeding, and these are referred to as oral cilia. So here's some figures of different ciliated organisms. Here you see cilia all around the outside. Over here, though, I know these aren't the best drawings. I'll get a better one here in a second. Over here, you're seeing cilia just around the mouth or the oral cavity of one of these organisms. And what they can actually do is kind of draw food inward to their cells. So here's a better figure of an oral cilia here. So this is an organism that would anchor itself to the bottom of an aquarium. And here you can see these little oral cilia that are around the opening. And what they do is they kind of flap and they actually draw nutrients from the water towards them, and then actually they stick to the cilia and then they can be absorbed for internal digestion. So that's another way that cilia can be used. Some organisms use them for one or the other. Sometimes they use them for both. So here you see some other illustration. These are actually all microscope images. These are all real images of ciliated eukaryotic organisms here. Okay, so basically they're just smaller flagella that are more numerous, and they serve, in addition to movement, also a function for filter feeding. Last but not least, we have in eukaryotic cells what can be referred to as cell connections. And there are a couple ways that they work, but they all kind of help to join cells together, which leads to the formation of multicellular organisms, but really it leads to what we refer to as tissue. So when we look at how multicellular organisms come about, they go through the process of individual cells joining together to make tissue, and then those going on to make organs and organ systems. Let's just show your cell, tissue, organs. There's a general flow here. So for example, you can see cells grouped together to form tissues, and then tissues go on to make organs and organ systems and an organism. But right now we're looking at how cells group together to form tissue. And the idea is they have to kind of stick together and attach together to form kind of a group of like cells. Here's another. I had it. I guess I lost it. Oh, here it is. This is a microscope image of some plant tissue. You see lots of cells all kind of joined together. So the way that they do this is with these cell connections, and they literally tie themselves, so to speak, together to form this clustering that we refer to ultimately as tissue. So there's a couple ways this works. One is called an adhesion junction, and this connects cells in a strong but flexible nature. So here they call it an adhering junction. And basically just between two cells, this is a way for them to kind of stick together. Another type, so this is kind of a basic connection mechanism there. Another type is a little different in that it's called a gap junction, or it's different in the sense that it allows things to travel back and forth, I should say. So these are called gap junctions. And what they do is they allow little tunnels in between two cells. And this is actually how substances can move from one cell to another within tissue. So this allows cells to kind of all have this kind of intricate connection system in which substances and nutrients can flow from one cell to the next. So that's called a gap junction. They allow direct cell-to-cell communication, as well as a variety of other things like nutrient sharing and things like that. The last type are called tight junctions. Tight junctions are kind of similar to adhering junctions, but the difference is they literally create a watertight layer between two cells. So anytime two cells want to mesh together in a way where nothing can really move between them, tight junctions are found. These are found, for example, in your intestinal lining, especially around the stomach, where you have gastric juice or gastric acid, like hydrochloric acid in your stomach. And this allows your cells in the stomach lining to be so tightly wound together that your stomach acid can't just easily leak out into the rest of your body. So tight junctions are similar but form a very, very tight waterproof layer between two cells. An example would be like in your stomach to keep the stomach acid from leaking out. Here was that same picture. Look at that. I forgot I had it there. But anyway, this is ultimately the first step in going from a single-celled type organism to a multi-celled organism. And it ultimately begins as one cell groups together to form a tissue, and that is based on these abilities to form junctions between each other. Okay, so that's kind of how that would lead to a more complex level of organization. Now, that's where you get into anatomy and physiology, and it kind of takes over from there to explain all that, the rest of that detail there. But from a microbiology standpoint, we leave it there, and we conclude here with Chapter 5, Part 1. Okay, we'll go on for Chapter 5, Part 2 here next, and as always, feel free to email me with any questions, and we'll take a look at the next part here in just a second.